Okay, hi guys, welcome to Biology Online with Henny van Vieren. Hey, year 12s, I hope you're really well and that you are still surviving the lockdown. I just thought I would record another quick video. This time it's going to be 2019 and it is question 1. So let's jump into that. So, as I said, 2019, question 1. So, here we have the fruit fly. It's Drosophila melanogaster. It displays a complete dominance. In other words, there's no co-dominance and there's no incomplete dominance here. Pattern for body color and eye color. The brown body color allele B, okay, is dominant over the black body color little b. And the allele for red eyes is dominant to the allele for brown eyes. So this one has got an dominant and dominant phenotype. This one has got recessive and recessive phenotype. So we can immediately say what that one's genotype is, can't we? So a biologist crossed a fruit fly homozygous for brown body color allele and red eye. Brown body is B and red eye is R. So if they are homozygous, it means it's got same. Okay, so state the with a fruit fly homozygous for black body and brown eyes, which you can see over there. So the gametes we know is going to be, if it's homozygous dominant, it's going to be big B, and big R for the mom, or then this one, and then this one is going to be little r, and little b. Okay, so there we go. Now they can only state the gametes. See, I didn't read my question. They didn't ask what is the genotype. State what gametes can be produced. And remember, if you are producing gametes, you're giving one of your alleles to the gamete, which in this case is going to be sex cells, which is sperm and or egg. Okay, so this one can only give a big R and a big B. So the gametes are going to be big B and big R. This one can only give little b and little r, so consequently that. Now the genotype of the F1 generation, remember these guys are the P generation, the parents, and then the first filial is the F1 generation, so that's the first time they mate. And so we have one that gives a big B, and one that gives a little b, and one that gives a big R, and one that gives a little r. Okay, and both of those are worth achieved points for you. So just like that, remember you only need 3 achieves to pass. How crazy is that? So just like that, you then have 2 of them already. Okay, so use the Punnett square below to show the gametes of the F1 generation and all the possible genotypes of the F2 generation. Okay, so remember this is our F1 generation. So in other words, they say use the Punnett square below to show the gametes of the F1 generation. So in the outside 4 squares, that's not part of this inner 16, that's where you're going to make the gametes. And how do we know what gamete or sperm or egg cells these guys can produce? We use FOIL, don't we? So firsts, and we write it in the first one. And then outside, which is big B, little r. I, so FOIL, I is the next one, inside, little b, big R, and then lasts, big B, little r. Bang. Okay, and we know that it's going to be the same because we're crossing 2 of the F1 generations with each other, so you can just take those and copy them down here. Alrighty, so now let's start over here, and if they're going to then create an F2 generation, we need to drag this across. Okay, so let's start with this one. So we're putting that across, and I'm putting spaces so that I can write the ones that's going to come down from the top. Okay, and that one, and that one, and that one. Okay, now I'm doing the same for this one, dragging them across, and then this one, and this one. Okay, so now this guy has now donated possible um, all the possible combinations of fertilization of two F1 gametes. Now I'm going to bring this down, okay, and I'm going to pull it down all the way. So that one goes, and we have big B, big B, big R, big R. If I bring this one down now, it's going to go big B, and big R, little r, and you know how this works. So I'm not going to go through that, but we can just do the first line. So big B, there we go. Big R, there we go. So big B, little b, and remember we always write the non-capital or the lowercase second. That's the convention. And then big R, little r. So there's the little r and there's the big R. And then the same thing happens down here. Okay, and that's how we fill them in. Now describe the predicted phenotype ratio. And the phenotype ratio is when we count them. So let's use a highlighter and we'll count all the ones that have, remember big B is brown bodies and red eyes. Okay, so whenever there's a capital B or a capital R, it's going to be a brown body or then a brown, but now we want that combination. So anywhere where there's a big and a big. Okay, and there's a big and there's a big. There's one there, and there's one there, and there's one there, and there's one there. And all of these will look the same, even though their genotypes are not the same, because remember we're doing phenotype ratio. And phenotype is the physical feature. Okay, so capital B, capital R. Oh, see, this one doesn't have capital R, so we can't count that one. Capital B, capital R, capital B, no capital R. So I have to not have that in there. Oops. Okay. Okay, here we have a capital B, capital R, capital B, capital R. No, 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 and yes. So let's count all the yellows. 1, 2, 3, 4, 5, 6, 7, 8, 9. Ha! Sounds so familiar. Okay, let's count the next one. So this is going to be then brown body but brown eyes. So there it is. So big B, little r. Big B, little r. And big B, little r. 1, 2, 3 individuals. Then our next next phenotype is going to be a black body and red eyes. Okay, so little b, big R, little b, big R, and little b, big R. There's another 3 of them, so we currently have 9 to 3 to 3. And lastly, we have the complete recessive phenotype. And we'll just make him orange. Okay, 9331. That was the expected or predicted phenotype ratio. So you have to write it out as 9 brown body red eyes, 3 brown body brown eyes, 3 black body red eyes, and 1 black body brown eyes. You have to write the words. If you don't have the words and only the numbers, you will not get your mark. Now, in this case, they were quite strict, so they only gave an achieved for all of that work and another achieved. But lo and behold, you only needed 3, so you have already got an A4 at this stage if you have all of those, which is really simple. Okay, so then we said wing length is another phenotype in the fruit fly. So you can see there, this one obviously has a much longer wingspan than its neighbor. So wing length is another phenotype in the fruit fly, and the gene is found on the same chromosome, this is interesting, as body color and eye color. Okay, I'm sorry, my voice is throwing a bit of a tantrum. Anyways, however, in contrast to the phenotype ratio for the F2 cross in part C, that's the previous answer, When the genes for body color and wing length are crossed, the observed ratio is 5:1:5. And the moment we see that, we remember that we learned that if you don't have the predicted outcome, then it is going to be due to linked genes. Now, I find it really interesting that they say here that body color and eye color are actually also on the same gene. You guys notice that? So, analyze the diagram below. So, eye color is over here. But body color is all the way down here. So if you cross over, there's a very easy chance that you're going to separate the eye color and the body color because they're so far apart. Even though they are on the same chromosome, there's a high probability that if crossing over happens, that they will be separated. Does that make sense? However, if you look at body color and wing length, they are very close to each other. Hence, when crossing over happens, then you have a very low probability that they will be unlinked, and that's why you get this weird ratio of 5:1:5, because that 1:1 meant that the chiasma for the crossing over was right in between there, and it's a very low probability of that happening. Okay, so let's look at the question further. Discuss with justified reasons why genes on the same chromosome can be both linked and unlinked, and we just looked at that. In your answer, you should include so remember, this is our main question. Our main question. If we don't take it back to that, we will not get our E's. Okay, you need to take it back to the main question. So answer these in relation to that. So in your answer, include a description of linked and unlinked genes, an explanation of how crossing over and segregation affects linked and unlinked genes. Now remember, when they ask you for an explanation of something, you always start with the definition. Always. Okay? And also, if they ask you to discuss something, you always start with your definitions. So a discussion of why body color and eye color are considered unlinked, whereas body color and wing length are considered linked. And there's our diagram again. Okay, so let's have a look at this. The first part is a description of linked and unlinked genes. So you can see here that first thing, this is our first A, we described a linked gene. Linked genes are alleles that are found on the same chromosome, therefore they are more likely to be inherited together. Bang, you have your A. Describe unlinked genes. Unlinked genes are found on different chromosomes and crossing over, sorry, spelling error, has no effect on whether they are inherited together or not. And then we describe crossing over. So remember, I told you that when they ask for an explanation, you always start with your definition. So crossing over and segregation. So we did linked and unlinked. So crossing over is the exchange of alleles or segments of a chromosome or DNA between homologous pairs of chromosomes. And I would have liked a better description there if you could say between the crossing over of homologous pairs of the non-sister chromatids. Okay, and that leads to recombinant chromosomes, or chromatids for that matter. Then during segregation, only one chromosome from each homologous pair. Remember, segregation is when we pull these pairs apart, not when you pull the two chromatids apart. That is the second part of meiosis. Okay, but in the first part, all we're doing is we're splitting the pairs, and that is going from diploid to haploid. Okay, and placed into the new gametes. Describe a gene. A gene is a section of DNA that codes for a specific trait of protein. 1, 2, 3, 4, 5 options there to get an A. And if you just studied the definitions, there is no way that you couldn't pass this, because you just needed 3 of them. Okay, discuss with justified reasons. So now we're going moving on to the explain part, okay, an explanation of how crossing over. But a better way when you're doing linked genes, we're going to start by explaining linked genes or alleles are found on the same chromosome. We know, we said that, therefore they are more likely to be inherited together. Genes that are closer together on the same chromosome are more likely to be linked than genes that are further apart. Remember, if you look at that and here, so we're just building on our previous answer where we're just going for achieve. So this is more towards merit level. Genes that are closer together on the same chromosome are more likely to be linked than genes found further apart because they will stay together during crossing over. And then unlinked genes are found on different chromosomes. Genes that are found further apart on the same chromosome can also be considered unlinked in some cases because they will not stay together in crossing over. Okay, so you're explaining now crossing over is the exchange of alleles or segments of between the homologous pairs of chromosomes. And with your previous section, you will get your M there. Crossing over can reshuffle or can create new allele combinations or new recombinant chromosomes. During segregation, only one chromosome from each homologous pair is placed into the new cells. Therefore, during gamete formation, alleles of each gene segregate, or the one that was separated from each other, so that each gamete carries only one allele per gene. Okay, so segregation is only one allele per, so that's the second part of segregation, is where we then break up these, and now you have only one allele. Okay, so that is segregation. So the first segregation, pulling them apart, the homologous pairs. The second part is pulling this as the chromatids apart. Segregation does not affect linked genes, as they will be inherited together or end up in the same gamete. Okay, so those two genes over here will be inherited together. However, segregation does lead to new combinations of unlinked genes, because remember, we have random assortment, and segregation is just going to put them randomly, split them into different cells. And explain, last thing you had to do was explain why body color and eye color are considered unlinked and all body color and wing length are considered linked, because they are so far apart on the same, and these ones are close together. That's all merit level. Now for your excellence level, then we had to do this discussion. So crossing over can separate linked genes. Linked genes occur on the same chromosome and are inherited together. Crossing over exchanges alleles between the chromosomes, between homologous pairs of chromosomes. Therefore, new combinations of alleles result. Look there. Crossing over exchanges alleles between homologous chromosomes. Therefore, the genes that are linked on the same chromosome can be separated if the chiasma, remember that's the point of crossing over, which is this point right there, if the chiasma is located between the two genes. Genes found far away from each other on the same chromosome are therefore likely to be separated via crossing over, due to the large distance between their loci. Remember, loci is the location of the gene on the chromosome. This means that they will be pulled apart via the process of segregation, and the alleles will end up in different gametes and will therefore not be inherited together. Although the body color and eye color, that's eye color and body color, although the body color and eye color are on the same chromosome, they are considered unlinked because they are found far apart and do not stay together during crossing over. However, body color and wing length are found on the same chromosome and close together. Therefore, they stay together during crossing over and are considered linked. There's your 2 E points. Okay, I hope that helps. That was the first question. Next time we'll look at the next question. Have a great day, guys. Bye.